Welcome to Neurosalience, the OHBM podcast.
Welcome to the Organization for Human Brain Mapping Neurosalience podcast. I'm Peter Bannattini, and here I discuss brain imaging findings and methods with scientists from around the world. Today, my guest is Rotem Botvinik-Nezer, a postdoc at Dartmouth University, working with Dr. Tor Wager in his cognitive and affective neuroscience lab. In 2020, Dr. Botvinik-Nezer was first author of an influential paper published in Nature titled Variability in the Analysis of a Single Neuroimaging Dataset by Many Teams, where the results were compared from 70 independent teams analyzing a single dataset having 9 hypotheses. This paper made it clear that there are many points of variability in data analysis pipelines and provided further incentives for sharing data and code to grow consensus and replicability. While the popular press suggested that this paper was yet another hit to fMRI, we discuss how even papers that critique the results of this seminal paper ultimately converge on agreement with the overall message of systematic transparency. Dr. Botvinik-Nezer also has a strong interest in how our brains influence our perception of pain, having just published a recent paper showing evidence that regions associated with painful stimuli remain active even when the subjects experience less pain while having the belief that a placebo is effective. In this conversation, we delve into all these topics and more, but spend the bulk of the discussion on the interplay between choices in analysis, such as determining the statistical threshold, and variability in results. We also discuss incentives for users to share data and code and possible ways to create a more solid scaffolding for best practices. Rotem was such a pleasure to have on as a guest. I hope you enjoy our conversation. Today, I'm with Rotem Botvinik-Nezer, and she's been doing some really outstanding work during her postdoc career so far. She first caught my attention when her paper came out that was published in Nature in 2020. Basically, it's the title of it is Variability in the Analysis of Single Neuroimaging Dataset by Many Teams. And it got a lot of attention, and it really helped push the field forward and make people a little bit more aware of the variability, even with the same data set. So thanks for coming on.
Thank you for inviting me. Yeah, it's great to be here.
Well, great. And so we'll get into talking about the paper and a few other, little bit of the work that you've done, and even in general, talking about, your perspective starting out as a young scientist and, being ready to move on to the next stage of more independence, and also your work with interacting with OHBM. But just to begin, I always like to start out, because I think everyone is always intrigued with people's backgrounds a little bit. Just in general, how did you, or was there any sort of specific occurrence, or was it a general, gradual movement towards neuroscience? How did you first become interested in neuroscience, and then specifically neuroimaging?
Yeah, so I was I think from a young age, I was very interested about biology and the brain and behavior. And then just before college, I read a book, Physiology of Behavior by Neil Carson, which is a book used in many classes in neuroscience. And I just read it, and it was so fascinating. I couldn't stop reading. And I think that's where I realized this is what I want to study. And I come from Israel, where you choose your major when you start college, not afterwards. So I just chose it, biology and psychology with emphasis on neuroscience. And ever since, I just, everything is so fascinating in the brain and the behavior. So I was cut off, and I just couldn't do anything else, I guess, from that point on. Yeah.
Well, that's cool. And so right. So you start, in the States that happens too, but people, are not, as far as picking their major, but then they switch a lot, but, or else they might, kind of try to hedge their bets and then figure out, what to do at some point midway through. And it's great, actually, the whole process of searching and reading these books and realizing, something, feeling when something resonates well with you. And So that's so you actually grew up in Tel Aviv, or did you?
Near Tel Aviv.
Okay, all right, great. Um, so you did your graduate work with Tom Schoenberg and Yanif Asaf. I recognize Yanif Asaf's name. He's uh, has done a lot of amazing work. Uh, what was that like, as far as that's concerned? Did you feel like this is what you wanted to do, and it seems like you shifted a little bit when you went into Tor Wager's lab?
Yeah. So I was always fascinated by placebo effect, like ever since reading this book, and in general, like all the mind-body interaction. So I knew I wanted to study placebo, but there was no real lab in Israel studying placebo in humans at the time, or focusing on that. And there were so many other topics that were interesting to me, so I just chose something else. I was during my undergrad, I worked with Yaniv Asaf and Ido Tavor and other people in his lab, and I did some plasticity, brain plasticity studies with diffusion imaging mainly.
Yep.
And I wanted to continue doing that in my PhD, but then I thought about the placebo and many behaviorals I want to study. And Tom Schoenberg just came back from doing a postdoc with Ross Boldrak and started his lab there. And Yaniv suggested that he's a great fit to me too. So I ended up doing a PhD with both. But actually, I was the first PhD student of Tom, so I kind of focused on with his lab, building with him. So I ended up doing decision-making studies with fMRI and diffusion imaging, and looking at the brain plasticity and neural changes when behavior change, and it was really great. Like I had so much fun, and I knew I just want to continue the research and hopefully have my own lab, which I'm lucky to be opening next year. So yeah, it was really great. Yeah, I mean, I have mainly positive things to say about all this time. They were wonderful mentors. So.
That's great to have good mentors who sort of open up things for you, and sort of show you various directions and opportunities, and so then you moved to Dartmouth. I mean, that must have been even in the States, going to Dartmouth, from people I've talked to, is a cultural. It's very different in the sense that it's a small town. It's a small college town in the middle of the woods and mountains. And what was that like, actually? Just
Yeah, so Tor was like a perfect fit for me because he's studying placebo, which is exactly what I wanted to study, and he's one of the board experts on that, and he's like using rigor, great methods that fit my interest, and he's such a kind person, great scientist. So I heard so many great things, and I knew this was probably the lab for me. Started talking to him actually before I came to Dartmouth. He was in Boulder, Colorado at the time.
Yeah.
So I have to say I did not choose the place. I chose the lab, but I'm probably a lucky human being because Dartmouth actually fits. I have a family and with kids, and this is a wonderful place for kids, and we love nature. So for me, this is much better than a big city, and I'm thrilled we were here. I mean, now it's been 4.5 years. I'm going to finish 5 years before going back to Israel. And I'm looking from my window now. There's a forest out there. I saw birds a few times. Like it's really great for us. So we love it here. But it's true. It's very different. Dartmouth is a smaller place, but there are amazing people in the department. You can learn a lot of things and many experts on different topics. So I don't know. For me, it was perfect, I guess.
Yeah. That's
also in the pandemic, by the way. That was a good place to be in.
Yeah, I mean, you're pretty isolated anyway, and so it's just sort of, and they have, I mean, I think that it's interesting. I remember I visited Dartmouth in right around 2000, the first time I visited Dartmouth for a workshop, and they were the first campus to actually have wireless internet on campus, like outside and everything in the courtyard. And I thought, this is just amazing. I can go outside with my computer and log on, and so that was ahead of its time in that regard. But yeah, no, I love the atmosphere of Dartmouth. I think it's just perfect for, getting focused. If you really just want to focus on the work and involve yourself with the great people there.
Yeah.
And Tor is amazing. And also I think as far as the group itself, and Dartmouth especially is strong in this and in other ways, is that you have a good solid neuroimaging core of people, and also especially with Tor is that he's a neuroscientist who cares about like a neuroscience effect, but he also cares about methods development.
Yes.
And that's really valuable. And it seems like you've been benefiting also from both.
For sure.
Yeah.
Yeah, he's an amazing scientist, and I'm learning a lot. And we have a large, very diverse group, so you can interact with people who know different things and have different ideas. And Tor is like million ideas per second, so that's really amazing to be here.
Yeah. Yeah, that's great. That's great. And yeah, already he's sort of like director. He's building his lab even more, and he's director of like the neuroimaging or the whole group there now. And that's just great. So getting into, why don't we just start with the paper? But just before we get into the paper, so it seems like a little bit of a shift in focus that you got into focusing on reproducibility or replicability. In this particular case, it's reproducibility. But how did you, like, what was your thought process in terms of thinking up this study, or even getting into this sort of area? Since it's like now you're into sort of like hardcore, processing and dealing with things like that.
No, that's a great question. So I think a few things that happened there. I always focus my research on the brain or behavior and asking questions about decision making in the past, and now placebo and beliefs and expectations. But I think at the end of my undergrad, I started to realize, that analysis and science in general is so complicated, and humans are performing it. So we have biases, and so many things can go wrong or can depend on, have different dependencies. So I started to think a lot about that, and while I was reading papers, and I think this combining with Tom, who came from Russ's lab and was very dedicated to open science reproducibility, I think it magnified it, my desire to focus on that, and the knowledge I had, and also papers were coming up about replication processing, project in psychology, and p-hacking, and all of this kind of stuff. So I read a lot, and I got very, very interested, and I just thought, there's a lot of things I want to do better in order to correctly answer or better answer the questions I'm interested in. So this got me to focus on that, and I should not get any credit for the idea for the Nature paper, because I was not part of it from at the beginning. It actually started from a group of economists who were part of the replication project in psychology, and then they came to Russ and suggested to do a project together about that, and he reached out to Tom, and then he started. I joined after the dataset was actually collected, and I called all the analysis part and all the teams and everything there. So yeah, they had a wonderful idea. I think it was very important, and the moment I got the opportunity to join, I kind of jumped on it, and it was just again, I'm a lucky person, like I said. So being interested in this stuff, and this opportunity came, and it was just a perfect fit. It was actually not part of my PhD, which was kind of interesting.
Yeah.
It's not part of my thesis, but I did it as at the end of my PhD.
Okay.
So, yeah.
That's interesting. It must have been. I mean, it's a huge amount of. I mean, obviously, recruiting all the groups to do this and sort of keeping track of everything, and it seems like it's a tour de force as far as everything that looked like it. That went into it as far as that's concerned. And actually, it's interesting that you mentioned that the other groups that were involved, because there was one aspect of the paper we could talk a little bit about it. It was kind of threw me a little bit when I started reading it. The whole concept of prediction markets, and we could get into that as to how that's a useful sort of construct in terms of getting a sense of how people believe the data is going to be correct or not. But we'll probably get into that in a second. So you had 9 hypotheses. You had 70 independent teams doing it, and it's called the NARPS dataset, the Neuroimaging Analysis Replication and Prediction Study. Just to, rather than me trying to summarize precisely everything, how would you summarize what your main thrust of the paper, what the findings were as far as that's concerned?
So we basically collected a new data set of 108 people performing a risky decision-making task, and then we distributed to 70 analysis teams. So each team is independent. It consists of 1 to 3 members. It could be a PI with a PhD student or like something like that. And each team was just asked to take the data along with 9 predefined hypotheses. So each hypothesis is about a specific brain region and whether it goes up or down with a specific aspect of the task. And then to analyze the data as they would for a paper in their lab. We did not want to impose any restrictions beside computing a whole brain analysis and not just an ROI one. And then report back to us. And we asked them to report 3 different things. One, just a binary decision for each hypothesis, whether or not it was supported by the data. Then a thresholded statistical map for each hypothesis, and then unthresholded statistical map for each hypothesis. And of course, all the methods they used. We followed Kobita's guidelines for that. And then what we did with that, we took it and we wanted to see whether results were variable and what the sources of variability could be and what we could learn from that. And what we found in general, I will start with the binary decisions. So in general, we found substantial variability there. I have to say that for some hypotheses, there was agreement. 3 of them were about the amygdala, and almost all teams said they were not significant. I think besides 3 or 4 teams for each one of them. But then for the other, and for one hypothesis, also like 86 or 87% said it is significant. But for the other 5 hypotheses, there was variability, where between like 20 to 30% of teams reported a different result than the rest, which I think was important because, it's the same data, the same hypothesis. They don't have any motivation to come to a specific, result. So you would probably expect a larger agreement, hopefully, right? So we were surprised by that, but then we wanted to look at the actual maps. And at the thresholded maps level, again, it was quite amazing. Like you could see very different maps for the same hypothesis and the same data. It could range from no significant voxels to like thousands or hundreds of thousands or tens of thousands of significant voxels.
Yeah.
And it got us, to really think about it. Because like you look at the map and you can't believe it's from the same data. But then in the unthresholded map level, I have to admit it was better. We tried to focus on that on the paper as well, saying that, the correlations between the different unthresholded maps were pretty high for most teams, not all of them, but for most teams. But the thing was that even for this, cluster of, I think it was 50 teams that had very correlated unthresholded maps, still they reported very different binary decisions. So, if they wrote a paper about that, the papers would have been very different.
Interesting.
And that's what he wanted to know, what happens in the wild, right? Because in the end of the day, you read papers and you want to know if the conclusions and the results you see, what led to them, and are they affected by the kind of flexible and, arbitrary decisions that people might have made. Sometimes you just do something because you're from a lab and this is how it's done there. Sometimes it's the default in the software you're using. Sometimes you did think about it, but you could come to a different, decision. So I think it just highlighted that we need to be mindful of that and think about how, the specific one analysis of one data set, what does it mean? What do we need to know if, if it's robust, and what could have been if something else was used in the pipeline, right?
Yeah. It seems that, in the paper you dissect this a little bit in terms of, there's many, many stages of analysis, from pre-processing to choosing your regressors to choosing your thresholds to denoising to, and then finally interpreting the results compared to, whatever you think should be there. And it seems that there's still a rich area for dissecting, what decision points, make the most difference. Have you looked into that?
Yeah, we did. So first of all, I forgot to say, the one thing that was really amazing, it was like 70 teams and no 2 teams chose the exact same pipeline. So for each team, there was a different pipeline. The things you mentioned, they could have used different regressor, different exclusions, different parameters in processing, different parametric tests or non-parametric tests. They could define the parcellations differently, right? So there were many differences there. We tried to look at the variability. We used a regression model to predict whether they reported significant or insignificant results. And we found that the estimated smoothness from the image was a significant predictor, and also the software that used FSL. People who used FSL reported more significant results, and people who used parametric tests reported more significant results. But I have to say, I wouldn't take these results and count on them too much, because 70 teams sounds like a lot, but it's not a lot of power to actually model all the different decisions, because there were so many. So we couldn't put most of them in the model. Just an example, where is the VMPFC, the ventromedial prefrontal cortex? Different teams decided, define it very differently, and there are so many options, we couldn't really add it to the model. So there were so many steps that we couldn't even model and test. So I think other studies are needed to really understand it. And great people in the field are now working on that. I could give examples like Camille Moumet and Greg Kier labs are working on that and trying to find the sources. So hopefully we could learn better about, if we want to see how specific decisions affect results, maybe we could focus on specific steps that make the most difference, right?
Right. Yeah. Yeah. Exactly. I think that I was reading through and I thought, well, it seems that to me, one other aspect is the choice of regressor. If it's what shape is it, what latencies, things like that, and that's obviously something. But there's many other things, and you're right. There's it's actually interesting. It's sort of a blind spot that everyone has. Everyone tries to do the most reasonable thing, and they think, okay, this is it. Everything that is real, I'll catch, and they don't think of, oh, well, if I, and we just don't have time to try, like, if I do this a little differently, how does this vary things? It's probably worth going through the exercise of that in some sense. And in this study, I think started the whole concept of, the multiverse or the of analysis. And I remember at OHBM the following year after this, I remember seeing Tom Nichols give a talk and showing all these, decision trees that sort of branch out and feeling overwhelmed in some sense, thinking, okay, what the heck? What are we going to do here?
Yeah.
On one hand, it felt restrictive, and also it felt like overwhelming, like we don't know what the right answer is. And what do you think is the way forward as far as, okay, so, well, before we even go there, I mean, obviously the popular press took this and said, oh, look, there's this study that says, fMRI results, even the same data set gives you very, very, very different results. And, there's other people, I was just mentioning before the podcast, and I'm co-author on this paper with Paul Taylor, who's been talking with you and collaborating, or not really, he basically wrote, well, if you report unthreshold data, it's not quite, things aren't quite as different as, if you have just these, isolated islands of thresholds. And everyone else, everyone tries to pick the right threshold, but everyone also kind of believes that that's somewhat arbitrary still, because we really don't know the nature of the noise perfectly, and there's all kinds of variables. And so, that's a question of like what the best thing to do is. Of course, you have to say it's happening somewhere, but then if you lower the threshold too much, things get similar, but that's not really a conclusion. It's sort of like a guidepost. But what's your take on that and how to go forward?
Yeah. Wow. So yeah, you mentioned so many things. I have so many things to say about it.
Yeah.
I will start by saying that I will say a few things. First of all, I think we need to combine many different practices for that. Like, there's no one solution that will solve everything, because a lot of things are here, right? So first of all, we should share our code, share our data, so other people can, use it to see if what happens if we change something, because you cannot really test yourself all the different options probably. And then this would help. Pre-registration would help if you know what you're going to do. So at least, like, there's, if you're doing one analysis, at least it's decided from the beginning and not based on the results that you see, which would help too. But it will not solve it, right? Because even a pre-registered shared analysis is still a single analysis, and we don't know what would have happened. So the multiverse is kind of a solution for that. We did not invent it. I think we just brought it to the neuroimaging world in a way. This term was specified years before, and in behavioral studies, people are using it, or specification curves when you show results on different pipelines, but usually it's for simpler studies. In neuroimaging, the multiverse is just so huge.
Yeah.
So many different decisions. And like what you mentioned with Tom Nichols, yeah, he's still developing methods to do image-based meta-analysis with the same data, because the data is dependent. You can't just use their usual methods. So he is doing great work on that. And we use some of it in the paper, in the NARPS paper. So the idea of multiverse is that let's just run many different pipelines and see if they converge to a single conclusion or not. But then there are many challenges. Which pipelines do you need to use? How do you test if they converge? How do you visualize it? How do you report it? How do you do it computationally? Right? If it's a large data set, it can be hard. But there are many aspects that different people are working on. Some I know of, some I don't know of course. But I think this would help a lot if we start doing that. It doesn't have to be 70 different teams. One team could do the same. Karp in 2012 showed he published a very important paper where he took a single dataset, fMRI dataset, and just ran almost 7,000 pipelines alone.
Wow.
And he compared the maps, and he saw that some of them were consistent across the pipelines, but some results really depended on the specific pipeline. So we did not invent the wheel. I think we more like tested it in real life, in the wild with real labs, just doing that. Because who knows how many of the 7,000 pipelines make sense to people, or people are actually using, right?
Yeah.
So the multiverse could help, if we find the right way to choose the pipelines and test for their convergence. And I think we're going to this direction in a way. I have to admit that for me, for now, I'm very mindful of that. So when I write a paper, I usually try to at least, test what happens with, major differences, like try something with a different test and then report. Of course, that's the thing, right? You need to report it. It doesn't help if you don't report it.
Yeah.
So I'm trying to report some differences in analysis and see how, if my results are robust to that, I think it's important. But there's still a lot of way to go. And what Paul Taylor and you and other people in this paper suggested is to highlight the results, the unthreshold results more, which I completely agree with. I think it's true, and the variability just grows when you go forward in your pipeline, right? When you threshold, when you look at a specific region, again, where is the VMPFC? Different people might say use different masks.
Yes.
Amygdala might be better defined, but still. So I think it would definitely help. I love the visualization that the paper suggested, and I think it would help. We just need to balance between, giving as much information as we can, but also giving something digestible, right, that people could build upon. There's so many data in the world, we can't just know all of it and conclusion based of it. So there's a trade-off, and I think if people also share their unthresholded maps, which I've always been doing, it helps too, because then people could use them for image-based meta-analysis, which is much better than coordinate-based. You just get more information.
Yeah.
So I think the combination of all of this would make it much better. But even the fact that people are more aware of it helps, I think, and reviewers are more aware of it, and you can think about, whether it affects results or not.
Yeah, it seems like there should be some other way than, so right, what Paul mentioned in his paper is that, essentially, if you unthreshold it and it was just sort of a salt and pepper sort of pattern or random, then clearly it doesn't do much value. It's not much value on thresholding, but it seems that there's still underlying, activity that seems to be lurking below the surface. And you think, okay, that if you average more, that would probably reach surpass the threshold. It seems that there should be some sort of method of iterating between thresholds and like sort of getting a better sense of the extent of the data without completely unthresholding. But I don't know. It seems that it's still evolving as far as it's concerned.
Yeah, I agree completely. Yeah, there's a long way to go before we set up on how to do it correctly. But there's great ideas out there. So.
Yeah. Also, so just briefly in the paper, you have you as I was reading it, I was just thinking, okay, now they're talking about prediction markets. I've never heard that concept before, and I was kind of curious: is that a useful thing to employ in other sorts of studies, or what was the goal of that approach, and what was it as far as that's concerned?
Yeah, so as I mentioned, the economists are actually the people who started this project. Felix Wolfmeister, Colin Kammer, Anna Dreber, Jürgen Jober, Magnus Johansson, and Michael Kirchler. It's a group of economists who were working together, and they did a prediction markets before. For example, when there was the project to replicate 100 studies in psychology, they used it, and the idea was to see whether peers can predict whether a study would replicate or not, and if they can. Then maybe we could, we don't need to replicate every study. We could use this knowledge to kind of predict what would be robust or not. That was the main idea. And the performance of the prediction markets was quite impressive in the first project. Here it was a bit, I will give some background. So in prediction market, what happens is that people, like researchers from the field, kind of go into a platform that illustrates a stock market. Yeah. Where each stock that you can invest in is kind of a study or a result, whether this study would replicate, for example.
Okay.
And then you get some money to start with, and you put your money, you invest your money, and based on your performance, whether you predicted the results correctly or not, you get paid at the end. So you have incentive to be correct. Yes. And as I said, in the previous studies before us, the performance was quite good, and people were able to predict the results nicely. Of course, not perfectly, but nicely. Here we could not do it exactly like that because we had, this one study but many teams. So the prediction was about how many teams would report a significant result in each hypothesis. So we had 9 hypotheses. And we had actually 2 different prediction markets. One was for people that were not part of the NARPS study, were not part of an analysis team. So they did not know, they did not see the data, they did not know the results, they just knew what we're doing. The other team was groups that were part of the study. They knew, they analyzed the data, they knew their own results. They did not know the results of anyone else. So at the time, the only people in the world who knew the results were me and Tom. And even not the economists who were in charge of the prediction market.
Okay.
So we had 2 different markets. We didn't want people who knew the data to affect people who did not or anything like that. No leak. And in both markets, what we found is that people were over-optimistic in thinking that, there are going to be significant results.
Huh.
So even groups that reported a non-significant result thought that most teams would say it's significant. So this was quite amazing.
Yeah.
And this study tried to replicate, previous literature. So they probably thought, we would be able to replicate these previous studies. Yeah. Another really interesting thing we found is that we asked people to estimate their expertise in neuroimaging and their expertise in decision sciences. So self-report. And there was absolutely no correlation between how their rate of expertise and whether they were able to predict the results correctly or not.
Huh.
It didn't matter if they were experts or not, if they were if they were young, senior or junior, nothing really predicted their performance. So this was interesting too.
That's interesting. And do you think the reason for that is it just, human nature, an intrinsic bias towards, being optimistic as far as the, seeing some of the literature and then having a sense of, okay, things are probably going to be, reproducible or, it's interesting. I mean, it sort of it taps into our own hidden biases.
Yeah, I think it's a combination probably of people are optimistic and, I don't know, I think when we read papers, like there's a bias where we think, so this is something we now know is true. So or think it's true. So we expect it to be true in the future as well. Yeah, definitely human biases.
Or do you think, or another way of potentially looking at it is, I mean, if you do lower the threshold, results get a little better, and maybe it's people's intrinsic ability to sort of, look beyond in some sense, or and have a sense of what's below the threshold and say, okay, that's probably going to be real or not. It'd be interesting to do it if you knew what ground truth was in some sense. If it matched, like, if it was it more optimistic than the results with the threshold results, and do you think, like, it would be interesting actually to lower the threshold just artificially, whatever, until it matched their predictions, in the sense that, and maybe is that like a crowdsourced way of of choosing thresholds. I don't know if that makes any sense. It just reminds me of other studies where people are, there's sort of a crowdsourcing of predicting, markets and other things, and usually, or like guessing someone's weight or something like that. Usually any one person is wrong, but if you have you average enough people, it actually usually ends up being true, whatever, more accurate. So I wonder if their predictions, I don't know. I'm not saying this is the case, but this is my own thought of maybe their predictions are accurate and maybe the thresholds were too stringent. I don't know, something like that.
Maybe it's definitely a possibility. I don't know. I wasn't I wasn't the investor of the market, but it might be. It's an interesting thought. I think we also need to remember that here they were predicting how many people would report a significant result and not necessarily how many people would reach the right conclusion. Because as you said, we don't know I don't know if the hypothesis is true or not in real life.
Yeah.
So that's another point to take into consideration. Yeah, I think also maybe people are just also used to seeing significant results because that's, the literature is full of them. So you kind of expect more significant results than not. And maybe the calibration of, wait, it's not a publication. That's like what happened in something. Maybe this was hard to, think about. No, because most of them were not significant based on the team's results. I don't know the true answer. Based on the team's results. So but maybe they were affected by non-threshold results. Mainly the teams, the people from the analysis teams.
That's interesting. So it was actually a nice added sort of thing that it might be worth doing in other contexts as well.
Yeah.
So, okay. So, and was there any, so as far as the feedback you got from the paper itself. So when the paper was eye opening and it sort of, I love papers like this because it sort of opens up blind spots in some sense. And, it gives you a sense of, okay, so we have to, this is a potential issue to deal with. And was there any pushback, or how did the field react in general to the work?
Yeah, so there were many reactions. I think in general it was very positive, and we also made sure to give the positive message, because we don't think, you said it in the beginning, the press can take it and say, oh, fMRI is invalid or something like that, but that's
Right.
not the case, right?
Yeah.
I mean, science is complicated. We just showed something here, and it's been shown before in behavioral data and the many analyst approach, and they showed it also in behavioral data. Things are depending on the analysis pipeline. So certainly here, when it's high dimensional, there are so many decisions to be made. It's also tested now in other modalities, EEG. It was tested in diffusion to some degree. So it seems the EEG results are not published yet, but it seems that this is true for many different fields, if not all. And I don't think it's unique to fMRI. That's what I'm trying to say. And the response were, some of them were a bit pushing back, but not a lot. I have to admit that most responses were positive, and 180 people from the field were part of that. So this also affected. Many people have been part of that, and they knew it before. They were part of writing the paper, so they were not surprised at this point, and also they felt part of it, and they were motivated, to test this when they joined the project. So I think this is actually a great example of how the field is, open to testing ourselves and finding solutions and improving. I think it's amazing. Like, it's really fun to be part of this field. And I was so lucky, to interact with all these great people, smart people, and learn from them and get feedback. And during the project already, think about what it means and what we can do. So I think overall it was positive and got us to, positive directions. There was some, articles saying something bad about neuroimaging. It happens once every few years, but it didn't really go to this direction in general, which is great.
I totally agree. I think that, and everyone I've talked to has said it's more of an eye-opening thing, and it's generally a positive slant that, and I do think that it sort of catalyzes ways forward. I mean, you've written other papers too. I mean, you just had one that was a little bit of a shift in this. So in Nature Human Behavior, 5 creative ways to promote reproducible science. And, it wasn't only talking about, the analysis, it's talking about sort of the whole infrastructure of how to change how people do science. You had things like funded project extensions for people who needed a little bit more time, or guaranteed funding for registered reports, and the whole concept of registered reports. So I don't know if you wanted to talk a little bit about that. And also, I love the idea of having a reproducible science knowledge base or a helpline and contact points and things like that. So that's actually sort of changing the culture of how things are funded or how people do it.
Yeah, completely. So as I said in the beginning of graduate school, I started, to focusing on this reproducibility and open science while doing, the psychology and neuroscience research. And I think what I did beside the paper we just discussed, the Nature paper, what I focused on the most is trying to help people, adopt these practices for reproducibility and open science. So, for example, me and Tom, my PhD advisor, we developed a course in Tel Aviv for graduate students where we taught them about all the open science practices and reproducibility and why we need them, the motivation and everything. It was really great experience for me. And then there are the papers that you just mentioned, I'll talk about in a second, and a few others that I've done, just trying to, help making it more accessible for people. Because I think as a researcher myself, and not one that, is focused only on the methods or writes the software themselves or something all the time. I mean, I write code, but I don't develop the tools myself that people are using in the world now. So I try to focus on, bridging between these, between all the great tools and platforms that people are making now to do open science and reproducible, and the actual researcher at the end that needs to use them and needs to do a lot of other stuff and know a lot of other stuff and have different, barriers. So this last paper that you just mentioned, the Nature Human Behavior Commentary, I did with Ronnie Tibbon and Josephina Wenroba from University of Nottingham, was just trying to think about creative ways on how we can help people use these tools. Of course, we need to change a lot of things systematically, but just try to, in the world we're living now, how can we do it better? And as you said, we suggested to fund projects better and to fund, for example, registered reports in a different way, maybe. And one thing that I focused on is the helpline that you mentioned. I think it would help if people will have like a centralized place where they can, get the knowledge they need in order to really start using these practices, and then they can see it's really easier than they might think.
Yeah.
And we tried to do this in a different paper that we published a year or two ago in Neuroimaging, where we just gave a review of all the reproducible and open science practices across the research cycle for neuroimaging specifically, and for all modalities, not just MRI. With a group of like 20 researchers from around the world. It was, again, wonderful collaboration and an opportunity to work with them. I led it together with Rotem Botvinik-Nezer and Yumar Nisa. And we also did an educational course about them in OHBM last year, where we just try to, give an overview of all the different tools in each stage. When you develop the idea, when you try to start a study, when you do quality control, when you analyze the data, when you test it, when you publish it, throughout the cycle.
Yeah.
And then just again, make it easier and develop kind of a resource where people can, have links to different resources, different tools, and they can use that. So we also have an online resource that the paper, based on the paper that people can use. And hopefully we could update it with time because, things keep changing. But I have to admit that this is one thing we're discussing in Nature Human Behavior paper, where because it's no one's job officially and no one gets paid to do that, it's hard to maintain it, because, like, for example, I'm moving now to start my own lab. I'm focusing on different things. It's hard to find the time to actually keep track and update things all the time.
Yeah.
So what we try to suggest is have some kind of a resource that, like we did in Neuroimaging, try to take a group of experts who can kind of start that and then find a way to make that work in the long term, to fund it. Maybe OHBM could, take responsibility for that in this case, for example, and have someone dedicated to that. Or maybe people can kind of pay it forward. They can contribute, get answers, and then contribute some information. There's the Neurostar forum, which is a great resource. But again, it's no one's job.
Yeah.
Which I think is a problem in a way.
Yeah. And I think that, certainly the NIH, they fund, they have educational grants and things like that for courses, but this is even beyond that, right? It's sort of having a resource that's constantly updated, that you can access anytime, that you can get training at or advice from. You can almost imagine, it's sort of a meta sort of science sort of funding that I think is important. People assume that with fMRI research or in brain imaging research in general, it's sort of organic. It's sort of like the methods sort of evolve and then they sort of settle in and it just works itself out. But they don't realize that, it could probably work itself out a lot faster and better if you had this sort of structure. And yeah, I like that idea. And who knows? I think that, something started by OHBM, but it's hard to actually, have funding agencies sort of think out of the box in this regard. But who knows? I think people are realizing, there's certainly a push to data sharing. And this is an example at the NIH, there's a big push to data sharing and everyone has to have a data sharing plan, but nobody really knows what the best practices in data sharing are. So they're already going, we have a data sharing team that we've started just for us, but everyone's going to this person, Adam Thomas, who, saying, hey, help us. And you can imagine something like that that would really be like an institutional knowledge of the field of best practices that evolves over time. I like that idea.
Yeah, we actually suggested both, like to have a centralized field-specific source, like what we started in neuroimaging and other people have done in neuroimaging. I think in neuroimaging we're in a good starting position if someone would be able to maintain it, but also have someone in the department, institution, or something that, because data sharing, for example, is very different depending on where you are. In Europe, there are different laws compared to the US, compared to China, compared to other places. So it helps to have both, kind of like someone close to you can just go and talk to. And it knows a lot about these things and about a specific characteristic in where you are, and also like a centralized source that the experts would keep updating, because new papers, new ideas, new tools are keep coming up. So really, I'm involved, and I have many friends there, and for me it's hard to keep up with all the tools. So I'm sure that someone who's not motivated or dedicated or familiar with this world, it's just how do you do it? How do you know what to use, what to do?
Yeah, and I mean, also, right. Speaking for myself, at least, I mean, in our group, all we do is try to come up with, better acquisition techniques, higher resolution. We're trying to invent ways of looking at, doing layer fMRI processing, and there's this Laney platform, and we haven't completely vetted it against best practices perfectly, and even time series analysis of extracting edge time series or windowed correlations and trying to make significant statements of that. It's hard to know. I mean, luckily we have people from AFNI or Gang Cheng who are good to consult with as far as what's the proper statistical method. But you're right, the methods are evolving very rapidly. And, even if you're trying to look at, transients in the signal and all kinds of things, people feel like, okay, so, what do you do there? You can't, it's almost, and there's this tension between setting up a registered, report versus exploratory analysis. I mean, and realistically, everyone kind of does both. Even if they do have a solid hypothesis, they still can't help themselves. And you mentioned this in one of your papers, how the idea of revising your hypothesis as you go along. I mean, at the extreme, it's just wrong. But somewhere in between, sometimes people do that, and then they say, okay, well, all right, let's do this new study with this different hypothesis, or obviously p-hacking is a problem too that I think we're made aware of too. So it's all these things are hard because it's messy with regard to human nature and pushing up against the limits of what's possible. And yeah.
Yeah, I think what I'm trying to do is I think the theoretical ideals are they keep changing in a way, but some of them are clear at this point. I think like what would be best to do or what would be most valid, but also there's the practical day-to-day life, the constraints that we have, limited resources, time, money, knowledge. I mean, you can't read. There's so many papers. You can't know everything, right? There's so many different things. So I think two things we need to take into consideration here is, first of all, that collaboration is really important, because if we combine our knowledge together, this will be better for sure. And the other thing is that we need to just balance between the ideal and the practical. So one thing Tor and I suggested recently in a book chapter that we was just published in the Sage Handbook of Cognitive and System Neuroscience, kind of suggested a phase model. It's kind of like clinical trials when they go through phases, And the idea is to just lower the barriers to begin for exploration. Exploration is important. We need both, right? We need both confirmatory studies and exploratory ones. We need to test hypotheses, but we also need to come up with new hypotheses, right? And learn new things. So we try to kind of suggest something that could balance between them, starting with studies that are more exploratory and then going up as more, studies with shared code and data, or reproducible studies, or studies that are tested for robustness to analytical variability. And then in different dimensions, there's pre-registration. So confirmatory studies that are shared or not shared, and reproducible and robust. So we kind of wanted to put it out there to see if we can kind of formalize the difference, and then each type of study is important and valid. We need all of them, but we can assess the evidence, based on how rigorous it was, how replicable it was, and how much we can trust it. But then we can do all of them. Yeah, we need to be mindful of that.
Yeah. So this was your book chapter, Advancing Cognitive Neuroscience, or was that the book, or is that the book chapter within?
No, that's a chapter. Advancing Cognitive Neuroscience is the chapter. The book is the Sage Handbook of Cognitive and System Neuroscience.
Okay. And yeah, I like your phase model. In some sense, it's sort of set up kind of like the idea of clinical trial phases or things like that, where, yeah, and you have the diagram laid out there. I like that concept. So the studies would be, like you say, in exploratory or confirmatory or shared exploratory, then reproducible exploratory, reproducible confirmatory, and then finally phase 4 is, robust exploratory versus robust confirmatory. So you have two parts of each phase, exploratory and confirmatory, and then the degree of certainty kind of goes forward with the phase. Is this sort of like how you should label your studies if you're doing them, or like in the paper, or how would you? Was this like how things are funded, or what's the plan as far as that's concerned?
That's a good question. Well, I don't have an specified answer for all of that. I think we should think about it more, and I think we need a group of people who, we gave like a preliminary suggestion, but we think that the actual phases should be defined by a consensus group or something like that. And yeah, it could be used. Each paper can kind of define themselves, and also like when you submit a paper, you could say which type it is, and then we could just. Again, it doesn't mean that phase 1 is not good or something like that. We need all of, and I think most studies currently are actually phase 1. If we're realistic about that. And we need people to explore. We need new methods, new ideas, new samples, but we also need to know to validate the results. I think we're not doing enough replications in neuroimaging. It's hard, you need money and time. It's just hard to do it. So I think it would give more incentive to do that, to replicate studies, to be more rigor, to share the code, to share the data, and then we could have results that we can kind of trust more in a way, right? Or tested more, and then we could, just proceed like that, and the literature could, correct itself better when we kind of easily know which type of study this is. But it has to be in combination of making it easier for people to do all these things, because we don't have usually millions of dollars per study like in clinical trials, right? And studies are complicated. In clinical trials, usually, one very well-defined test, simple usually, one analysis, and so many people are involved, so much money, documentation. So we can't expect that for every study in basic science. It won't happen.
Yeah, it's still very dynamic and sort of organic, and but yeah, as I was reading this, and I mentioned this to you before, as I was reading this this chapter, sort of skimming it and looking at this diagram, I thought, well, some journals, and so this is one thought too. It's like some journals like to have the idea of having like living documents that sort of are updated or whatever. But instead of that, and I mentioned this, is that maybe there could be funding for some group of people who makes a database of the papers and sort of give them a set of metrics, like has this been reproduced, or some value of like how rigorous the analyses are, how well is the data shared, is the code shared, sort of having each paper have some sort of an overall set of quality metrics that how impactful it might be in the field. I mean, and they could just be totally separate, just a database, and could just look up the papers. And I don't know, it would be interesting to see if that would get any traction, because everyone wants to know. I mean, otherwise right now it's like you read the paper and you're like, oh, it seems pretty good, and it's like you don't really know. You'd have to do some work in terms of judging it. If you have a team of people focused on just scouring the latest literature and sort of going over, and it might open up possible opportunities, or it might help change the culture of the field.
Yeah. I think it's a cool idea. I mean, the funding needs to be realized somehow. I think a lot of the problems are the funding, right, and incentives, and how you get credit kind of for what you do. And also, I think another thing that would need to be considered here is that it's kind of subjective sometimes. How rigor, there's no one clear answer about how good the paper is. So we need to be careful of that. Like, I don't want to just judge papers. I do want to get information that's important, and some things are objective, like if the data are shared or not, if this is objective, for example. Was it registered? I mean, it's objective, but also ambiguous to some degree, because there are different registrations, right? There are different degrees of data sharing, so it's not that like black and white. But yeah, I agree. Like, knowing all this stuff could help, and having some kind of a resource for that. Actually, I was in Neuro Academy in summer school in 2018. Neuroimaging and open science, and in the hackathon we suggested something that was called an O factor, which is an open science factor for journals. We started to like code something for that. We never really completed it, but like everything in most hackathons. But the idea was that like was try to find, not that exactly, but I think quite close, like try to give kind of, instead of the impact factor, give another metric for journals of how open and reproducible they are, and then to papers and maybe to people or to labs. Or it didn't reach that, but the idea was to try to give credit for people for doing this thing, because at the end of the day, you make a lot of effort to share your data, Do you get something? I mean, you should do it because you're motivated to do it.
Yes.
But also, people should acknowledge that it's hard and that people are working hard to, collect the data and share them and make sure things are reproducible. It takes much more time. But if we want to get to the true right answer, we need to do that because, it takes time, but it makes it better, the science. So we need to incentivize that. I think it's more important to incentivize than to, enforce it in some way.
I totally agree. Yeah, and you're right. And you can't just rely on people's wanting to do the right thing. Everyone has limited time, and right. It's all about incentives, either positive or negative. But one of the, a little bit of both would help change the culture, I think.
Yeah.
Yeah, that's actually a really interesting problem. I think OHBM probably should focus more on things at that level of how to incentivize this more. So, okay, I mean, there's so much more we could talk about with this, but it's also it's also interesting because in terms of changing the culture, just to finish up this, there's still people who I talk with from time to time who think, well, the whole natural process of papers being accepted and it's either the papers die out or they don't die out based on how real the results are. People feel like, okay, that's probably it's been working for the last 100 or so years. Why not just continue it? And I think that while that's true, it does work. It's just slow and inefficient. And it seems that now, as things are accelerating, it's all the more important to have better structures for that. So,
I mean, I agree in general, but I think also I'm not sure it's working well now because I think the amount of papers just so it's exponentially growing, and the papers will survive or not might not depend on whether they're true or not, but also whether what lab they came from, who wrote them, how exposed they are in social media, how much they gain traction. There's some other reasons for paper to persist, although it might be wrong.
Yes.
And again, because in neuroimaging, replications are rare, I don't think we're really testing. And I know personally of a lot of examples of people who try to do something they were sure would gonna work just because papers are published on that. It didn't work, and then you just do something else. You don't really publish, actually that it didn't work. So I think that's the issue. Humans are doing science, so we need to keep that in mind. And humans are not objective, and humans have biases, and they affect what we publish at the end and what we understand from what we read. So I think just trusting on that happening on itself, I would be careful with it.
Yes, no, I agree with you, and you're right. It does break down when the system gets too large, and you just don't want the field to be spinning its wheels. And that's there's probably a lot of energy just spent that way. And like you said, there's a lot of there's so many negative results that are not reported, and people try those experiments, and then they can't find them. They don't report them again, and so it's like so much things like that. It would be worth writing about all the wasted effort. Review article of all the wasted effort.
And money,
And money.
We have limited resources. I think we should use them wisely. And I would like to, invest them on things that are more likely to work or really worked in the past, and not, work for one person but not for 100 that I'm not aware of. Right?
Yeah.
So,
yeah. It's okay. Well, how about like I said, there's so much more to talk about, but I realize I just wanted to try to talk about two other things really quickly. You actually do other work other than caring about meta-analysis and replication, and you have a paper right now. It's in bioRxiv. I believe it's on
under review.
Okay, it's under review. Placebo treatment affects brain systems related to affective and cognitive processes, but not nociceptive pain. And it was really obviously this is sort of along the theme of what Tor cares about, but also what you're interested in. So this was eye-opening in some sense. This was one of the first papers I don't follow the literature closely, but it seems that looking at the brain activation associated with placebo effects and showing how there's no change in like your typical pain centers, but there are changes elsewhere. So why don't you talk about it a little bit there? It was very cool.
Yeah, thanks. Yeah, so as I said in the beginning, what I really wanted to study always is placebo, and I'm opening my own lab in the summer, and that's what I'm going to focus on: placebo and expectation and beliefs, and how we update them. And so all the reproducibility, open science things are a major part of what I do, but they're not really my focus in a way, right? So Yeah, what we're trying to do there was to take placebo. So placebo effects are studied in many different labs, and in the last decade or 2 decades, it's gaining a lot of attention. And we're trying to understand what's the mechanism, why placebos are working so well, and maybe how we can leverage that to improve clinical trials and clinical practice, treatment in general. So what we did in this paper, we used a very large sample size for for a test like placebo. It was with almost 400 people. I know it doesn't sound a lot compared to biobank or something, but it's a very specific task for one study, which is a lot.
Yeah.
And we tried with this large sample to test previous results and a question of whether placebo in analgesia, whether it affects the pain nociceptive regions, like the regions that responds to the pain in the lower level, or the higher level interpretation that we give. Because pain at the end, the pain that we report or feel is the combination of the nociception and the interpretation that we give it, emotional, social, cognitive aspects. And there were inconsistencies in the past, as many studies have shown effects on lower level things like in the spinal cord and on insula and thalamus and lower level regions. But we want to test it with a large sample size and see what happens. And what we found is that actually these lower level brain regions, like the pain metrics, it's called, the regions of response to pain, were not affected by the placebo. And they were not affected. We tested because of the large sample size, we used Bayes factors, which allows you to test for evidence in favor of the null effect, not just in favor of the H1 hypothesis. So we found significant null effects, right? Strong support for null effect, meaning that placebo does not affect these regions, or neuromarkers that represent the response to nociception pain, but rather that higher level regions are affected. Prefrontal cortex, striatal regions, and a neuromarker that was developed before. And I think what's unique in this study is, again, we had the large sample size. We also pre-registered specific neuromarkers and regions that we were about to test. So the exploration was constrained, and that we could use the Bayes factors to give. I think it's a good direction in general to try and provide evidence for null versus alternative. Just because it's not enough to know if something is significant or not. It's also, if it's not significant, it's interesting if it's if you can learn that it's just not different or something like that, and not just that it's not significantly different, right?
Yeah, could you explain a little bit more how you do that? Because I think a lot of people still just have in their mind that you can never disprove an hypothesis. So how did you set up the null when you're trying to prove something doesn't change?
Yes, so we never prove, right? We cannot prove that. I agree with that. But what we do is we kind of quantify the evidence in favor of the alternative hypothesis compared to the null hypothesis. So you need to define both hypotheses. You have some priors because it's based on statistics, but there are some default reasonable ones that you could use if you don't have a specific direction. Yeah. We use neutral ones here. And we also tested, just going back to the analytical variability, I tested with different priors to make sure my results are robust to that. So this is one thing how I kind of apply what I learned from NARPS to my own studies. Yeah, I guess. So they're just a way, the Bayes factors just help you get a number that says how much the evidence supports the null compared to the alternative hypothesis. And different numbers provide weak or strong support for one of them. You need large sample size to really get strong support for null, like to be able to get strong support for the null. But we had it here. So we got very strong support of the null, and we learned a lot from that. I think the field placebo could learn a lot from that. Okay. Yeah.
Okay. So in terms of interpreting this result, so did you actually look also at everyone responds to placebos in different ways. The individual subject variation and or even like the behavioral response as it related to the other areas that might be essential for engaging the placebo sort of effect. So those areas didn't go down in terms of the nociceptive areas, but you had mentioned there might be other. What do you think is what do you think's going on as far as how we do suppress pain by our belief that something's working, and different people have different skills in this. Was there any variation with people's skill or belief?
Yeah, so there's a lot of individual differences for sure.
Yes.
And people are trying to predict who would respond to placebos. There are many studies on that. We did not focus on that on this specific paper, but we have a lot of data, so we're going to.
Yeah.
But in general, what we think is going on is that, again, pain, there's like a model of pain that says that like pain is a combination of the biological, psychological, and social aspects. You send something in your receptors, it goes up to your brain, and then it combines with some top-down processes of the context, what you expect would happen, what you believe about it, who is with you currently, what do you think about the physician who may be treating you, and a lot of other characteristics. And all of this together combines to give how much pain you feel.
Yeah.
And of course, there's another step of how much pain you report. It doesn't have to be the same, right? Maybe you're trying to show how strong you are now and how resistant you are, and you say something else. But what we found here is that it wasn't a decision bias. Like, it wasn't like they felt high pain, but they reported less or something. But the interpretation, the higher level, the top-down processes affected how they, how much pain they actually felt
Yes.
at the end of the day. And then the top-down regions are kind of interacting with the lower, with the bottom-up signal, and together give the final result. So, I don't know exactly the specific mechanism yet, but I think it's definitely something that goes from top down, and it does not change the lower level. It doesn't seem to change the lower level processes. But also we tested, a specific type of placebo in pain, and with thermal pain and mechanical pain specifically. And we use suggestions and conditioning. There are many different types of ways to induce placebo, and it could work differently for different people and for different contexts. So this is much more complicated than that. But I think it's a start, to give us some clue of where to look.
Yeah. It's actually, it reminded me when I was reading this a little bit of a very early study back in the early 2000s, one of the first ones. I don't even know if that was replicated well, but of neurofeedback, of basically, you identify the regions. At least here, the study just identified the main regions that are associated with pain, and they had subject, those areas were fed back to the subject, and they came up with a strategy for reducing the activity in those areas, and it corresponded with them feeling less pain. So while I don't know about, like I said, the quality of those results, there seems like there's a link in terms of, once you identify those areas, either you could, you could do neuromodulation, or you could, give feedback, neurofeedback based on those areas to either increase or decrease, and it might have behavioral effects. But yeah, no, it's a really interesting area. I mean, it's wide open in terms of your own ability of your mind to affect your subjective experience. And it seems like couldn't you also measure subjective experience of pain by things like skin conductance, or it's like instead of just using the report. I mean,
Yeah, definitely. Yeah, many studies are using skin conductance specifically. We did not analyze it here, but what Tor and us are trying to do is to develop neuromarkers that could, serve as the objective measure, and there's the NPS, neurologic pain signature, which we use in this study too, which was validated in many studies now, and it's like a neuromarker that responds to changes in the nociception. So if you give like lower intensity stimulus, for example, it will respond less. But if you do all this top-down interventions, like cues that make you expect less pain or placebo, it seems that it's not affected by that. So it is more like of a signature that of the nociception part of that. And there's another signature we've used, it's called SIPS, which is the other part, like the higher level. So we're trying to use the brain as this, biomarker for pain or for placebo effects. And I think it's a really promising direction because, we should not just rely on self-reports, because, as I said before, it can also be affected by decision biases. And also maybe there's some people who can't report because they're like in clinical conditions that are not allowing them to report pain, and we want to know if they're in pain. So it would be very beneficial to be able to know that without talking to them.
Yeah. Okay. Yeah. No, as you're saying this, I recall now suddenly that Tor had this pretty amazing paper in the New England Journal of Medicine on objective measures of neuro measures of pain. And
that was the NPS paper.
Yeah.
I think in general placebos are very promising. I mean, people see them as like, no, we're tricking people or deceiving them, but I think what we learn from placebo is that there are like effects there that are just your own body kind of healing yourself, and we could just use it. We don't need to deceive anyone. We could understand them better and use them with active treatments, and make people better, right? So I'm very excited about that, like about trying to develop ways to do that.
Yeah, that's great. If they just make it into a science of sort of, have a clear mechanism by how that's done. Yeah, that'd be great. So, okay, well, that was a really, really cool study. Are you going to, so let's talk a little bit about moving on with your career, and also, OHBM. Well, the first question that comes to mind from this, though, will you be continuing your studies? So you'll be moving to Jerusalem, actually. Do they have neuroimaging, or what are you planning on doing?
Yeah, of course. So I'm going to open my lab in Dartmouth University this summer. They have MRI machine, Prisma machine. So we're definitely going to continue doing some MRI studies of MRI, along with computational behavioral studies, studying placebo and expectations and beliefs, how they change, and hopefully trying to leverage them in clinical practice better. And so OHBM, it's kind of where I started. Like OHBM was my first international conference, and in Geneva, it was great, and I had so much fun. And ever since, every time I went to OHBM, I really enjoyed meeting with all the friends and colleagues and mentors there. And they're so great. I really feel part of this community, and with NARPS, collaborating with over 180 people from there. It was just amazing for me, and I'm collaborating with many different people. So I'm just having so much fun. I was also in Montreal this summer, and I think it was a great conference. Fortunately, I don't think I will be able to come to Korea this summer because I'm just moving back. But I'm gonna miss it this year probably. But still, I think OHBM will always be the organization I feel like I belong to, and I want to go to, and I want to send my students to.
Well, I think. I think actually, I'm glad to hear that, and I do always feel that OHBM is very much fostering of young scientists who also, and it's nice in the sense that they care about the brain, of course, but they care about methods, and they just kind of straddle those two, and really help people in terms of figuring out, what the best practices are. And it seems like I do get that sense where the culture is very collaborative. They care a lot about doing things right, and I think you fit in perfectly as far as that's concerned. That's great to hear. And it keeps on evolving. I think that, I think at least half the people at OHBM now sort of are doing some sort of a meta-analysis of other databases and things like that. So it's very much becoming even more open science, and they're evolving the field that way. It just reminds me also. You wrote a piece on virtual conferences, during the height of the pandemic, and how to make them more inclusive. And I thought that was really a nice sort of summary of everything. And unfortunately, OHBM is trying to do that, but it's so expensive to do it right. And that's everything is sort of comes down to how do you actually lower the costs of these things?
I agree. I think most of the credit for this paper should not go to me. It's the Open Science SIG that I'm co-author, and I helped, and it was great. But the Open Science SIG at the time of COVID was in charge of that. I think it's definitely a challenge because, after a few years of, participating in a few online conferences, even if you really try to make it it's inclusive. You can be there, but it's just hard it's not the same. At the end of the day, the in-person experience is just not the same. So I think we will have to just have a combination in a way, right? Hopefully people would be able to come to the in-person component, but it would help to record, and OHBM is doing that, right? To record the talks and make them accessible for people who could not come or who came but missed the lecture or were there and want to hear it again. So I think OHBM is navigating that quite nicely. It's not perfect, of course, but I haven't found the perfect solution too. So it's just, I guess we will just continue to make it better, hopefully, and hopefully the prices will go down at the end when technology,
I think inevitably, I think it will, and you're right. And I think there's other ways of non-real time. You can always, and I think the community is such that you can always email anyone or contact them, and they can give you their data. Or there's these databases and code, and it's like it seems like it's becoming more of a even if the conference itself is not in real time, virtual, all the information is accessible becoming more accessible thanks
Yeah.
to things like this, what you're doing.
That's important, yeah. And people are, and as you said, like the field, I think the neuroimaging field is really one of the most advanced ones in like open science and sharing, and we're really like making a change. So I really feel fortunate to be part of this field and belong to this field. So I think we're going through great directions. There's a lot of work. There's a lot of barriers, but
there's barriers. There's cultural barriers, and things evolve as they do. But the leadership keeps on expanding and growing, and I think it's a really robust society that also feels like a family, and you just don't have that, I think, in other types of scientific communities, and I think it really fosters young scientists. A lot of times you have where the older scientists sort of are there and they sort of dominate with their labs and whatever, and certainly I actually feel that there's much more of this sort of bottom up upswelling of energy at OHBM. So that's great. Yeah, and thanks to your work and everything like that, I think that helps foster the whole infrastructure. So
thank you. I'm a small, very, very small part of that, but I'm happy to be a very, very small part of it.
Yeah. Okay. Well, I think that pretty much covers most everything. Yeah. I mean, there's obviously so much more we could talk about, but yeah, thanks for coming on, and I wish you the very best moving back to Israel, and definitely, hopefully, keeping engaged in OHBM.
Yeah, thank you so much for having me. It was super fun. I'm looking forward to doing it. Yeah.
All right. Well, thank you.
Thank you.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Xiqian Michel Li and Ömer Faruk Gülban.